a slight reduction in the levels in Northern America and Europe. If we also analyze the food insecurity in the world, we can see that more of 2 billion people suffer from moderate of severe food insecurity, and this proportion compared to the global or the total population is considerably higher in continents such as Africa. Another important factor affecting the human nutrition currently is the hidden hunger or micronutrient malnutrition. The hidden hunger affects approximately 2 billion people, or 1 in 3 globally, and is characterized by a chronic lack of essential vitamins and minerals, such as vitamin A, iron, and zinc. 20% of maternal deaths and 45% of all child deaths are attributable to undernutrition. In fact, we can even suffer from hidden hunger because the diet and the food we consume currently doesn't have the enough content of micronutrients. This hidden hunger has different effects for human health, for example, cognitive impairment, weak immune system, maternal and infant mortality, vision problems, fatigue, weakness, and reproductive problems. In this scenario of malnutrition and food insecurity, common bean is a promising commodity to ensure food security in the world. Common bean is the most cultivated legume crop in the world for direct human consumption, with a global production exceeding 30 million tons in 2018. Dry beans represent an important source of protein, dietary fiber, nutrients, carbohydrates, vitamins, mainly in developing countries in Latin America and Africa. In the case of beans, natural genetic variability for different traits, including quality traits such as nutrient and protein content, has been reported. And this genetic variability offers a scope for studying the genetic basis and developing cultivars with better nutritional profiles. Common bean has two major ecogeographically and genetically distinct wild gene pools. The first one is the Mesoamerican or the Middle American gene pool, which is located in Central America from Mexico to Colombia. And this pool has different accessions with features, for example, colors, with different colors, sizes, etc. The second pool is located in South America and is the Andean pool, located in the Andes. And also, we also have different characteristics, for example, pigmentation that is different in the accessions. There is also a third and minor gene pool, which is located in Ecuador and Peru, and is constituted by wild populations that only grow in these specific areas. And the distinctiveness is the presence of a very specific type of facilin that hasn't been found in the other two gene pools. Considering the potential of beans and the variability we have in this crop, my thesis proposal is based on two projects or two main ideas. In the project one, as part of the improvement efforts in different crops, different technologies has been used to test, in the case of beans, the gene pools and domesticated races in order to understand the genetic basis associated with different traits. One approach is the genome-wide association studies, GWAS, where we take advantage of genotyping and phenotyping in an association panel. An association panel is a population of diverse genetic lines and only needs to be genotyped once, but we can rephenotype them for many traits in different environments. In this case, for the genotyping, we use different molecular markers. Some of them are the SNPs, or single nucleotide polymorphisms, that we can get using SNP array chips and imputation of data, or whole genome sequencing or resequencing depending on the species. In the case of the GWAS associations, we correlate the different molecular markers and their genomic positions with the phenotype in order to get p-values and identify molecular markers that are significantly associated with the traits of interest. These associations have some limitations because some SNPs that we can identify that are associated are usually located in non-coding regions, in intergenic regions, or intronic regions. To overcome these limitations of GWAS, and considering that also differences in the abundance of transcripts of genes can lead to differences in the phenotype, a second approach has been proposed, which is the expression quantitative trait lossy, or EQTL. In EQTL, we used genotyping and RNA expression data. We extract RNA from different tissues at different moments, depending on the interest of our research, and we analyze the level of expression of genes using one or of two approaches. One is to analyze the single level of expression of different genes that are involved in the trait of interest. And with the advance of next generation sequencing technologies, we can also have RNA-seq data. In this case, the EQTL analysis treats the RNA expression levels as quantitative traits, and we can correlate the molecular markers and their genomic positions with the expression data in order to identify SNPs that are not directly related to the phenotype, but SNPs or markers that are associated with changes in the level of expression of genes that are involved in the trait of interest. In this case, we can, for example, identify the effect of different genotypes in the level of expression of genes in order to identify which one is which genotype is the genotype that increase or reduce the expression level. As a disadvantage of this approach, despite the many advantages we have, is that the evaluation of local EQTL in the context of complex traits usually includes only the lead variant of a phenotype-associated locus, as linkage, disequilibrium, and haplotype structures complicate the analysis. So to complement this EQTL analysis and refine the analysis, a third method has been proposed recently, which is a transcriptome-wide association studies. In this case, we have two main approaches. In the first one, we used phenotyping, RNA expression, and genotyping in different steps. First, we perform a GWAS analysis, as I explained before, and we identified markers that are associated with the trait of interest. And then we input data for transcriptome, and then we test for association in order to identify genes that are positively or negatively associated with the trait. This is one approach that has been used in plants and mainly in medical applications. And the disadvantage that some authors mention of this first approach is that this one is based on summary statistics, LD scores, and the use of indirect data with the imputation. So a second approach for the TWAS is using phenotyping and RNA expression in order to correlate them directly. So here we can correlate the RNA expression data for different genes, and we can identify single genes where the level of expression is associated with the terminal phenotype. And in this case, we will identify these single genes. As summary of this first part, we can see that depending on the level of analysis that we are trying to make, we can differentiate different analyses. For example, if we are considering DNA and the phenotype, we can perform analysis such as GWAS, linkage mapping. But if we are using DNA and RNA data, we can perform EQTL analysis. But if we want to use RNA and the phenotype, we can use TWAS with any of the two approaches that I explained before. The second project, or the second idea, is based on mutations. As we may know, the mutations occur naturally as part of the constant influx of genetic variants into populations. These mutations can be in the DNA, for example, substitutions, deletions, or additions, and according to their fitness effect, the mutations can be deleterious, neutral, or advantageous for plants. Strongly deleterious mutations are purged from populations by purifying selection, and strongly advantageous mutations increase in frequency and are fixed in the populations. However, there are some weakly deleterious mutations that have the potential to persist in populations and remain hidden from the purifying selection. This kind of mutations and variability is present among natural and breeding populations. In the case of selfing species such as common bean, these deleterious mutations are usually exposed and purged from the populations, but the effective recombination rate also impacts the number and the distribution of deleterious mutations in the genome. In this case, genomic regions with a low recombination rate are prone to have a higher number of deleterious mutations that remain in the genetic pool of the population. These kind of non-synonymous variants that have deleterious effects can reach percentage of 40% in some species such as yeast, and in the case of plants, for example, rice, maize, and Arabidopsis, the percentage can be the 20% of these kind of variants. In addition to these kind of deleterious mutations, during the selfing process also can appear different spontaneous mutations. So the study of the purely purged and spontaneous mutations offer alternatives for crop improvement, aiding to their identification and the elimination to reduce the restrictions for genetic gain in breeding programs. To identify these mutations or to set apart what the good mutations or the bad mutations, there are different approaches. There are two main approaches. The first one is the quantitative genetic approaches, plus mutation accumulation studies, where we estimate the mutational effects on fitness of the plant, and we can also integrate DNA sequencing data. The second approach is based on different algorithms, where we used sequence conservation analysis and DNA sequencing. Some of the algorithms that are very common are, for example, SIFT, LRT, and polyphen2, that are sequence conservation approaches that don't use phenotypic data. So taking into account this background, I have some objectives for each project. For the first one, the objectives are to identify marker trait associations for quality traits in common bean, to detect markers involved in the expression control of genes related to quality traits, and to associate gene expression changes with final phenotypes for quality traits in dry bean. In the case of the project 2 that is related to de-novo mutations, the objectives are to identify the role of different mutations and their effect on fitness of a common bean population, to establish a relationship between the recombination rate and the presence of putative deleterious mutations in common bean, and to determine the best approach or algorithms for the identification of deleterious mutation in common bean. As hypothesis, I have two main hypotheses, one for each project. The first one is that there are genetic variants at the DNA and the gene expression levels that modulate the quality traits in common bean that can be robustly identified by using different approaches simultaneously. And the second hypothesis is that common bean populations with a high level of homozygosity harbor pre-existing and de-novo mutations that are potentially deleterious and have effects on plant fitness. So to reach these objectives, I have different materials and methods. For the project 1, here we can see an overview of the methods from plant material, genotyping, phenotyping, expression data, and data analysis. For plant material, I will use a Mesoamerican bean panel. And for the genotyping, this panel was previously genotyped using a SNP array chip with more than 5,300 SNPs. In the case of phenotyping, I plan to measure some variables related to quality traits, for example, nutrient and protein content, anti-nutrients, digestibility, and aroma. For these variables, for example, in nutrients, I will use acid digestion method and the quantification using inducively coupled plasma mass spectrophotometry or inducively coupled plasma optical emission spectroscopy depending on the available equipment. For crude protein content, I will use the Kjeldahl method for digestion, and I will use the factor of 6.25, where the content of protein is 6.25 times the content of nitrogen in the sample. For anti-nutrients determination, for example, for phytic acid, I will use the weights reagent, which is a colorimetric method. For trypsin inhibitor activity, I will use the Kadicki method using benzoyl DL-arginine for nitroaniline hydrochloride and dimethyl sulfoxide. And for tannin content, I will use spectrophotometry at 760 nanometers using Folin Denis reagent and extraction with 1% of hydrochloric acid in methanol. For in vitro protein digestibility, I will use a multi-enzyme method using pH measurements of different enzymes and controls. And I will also test a quality trait that is the aroma, which is a valuable trait for the market. And I will use fresh immature seeds and dichloromethane extraction and gas chromatography mass spectrometer with silica capillarity columns in order to identify the compounds that are changing the aroma of different bean samples. Finally, the expression data, for the expression data, I will get RNA expression or RNA-seq data. And for the data analysis, the idea is to combine the different approaches I explained before. In the GWAS, I will use a mixed linear model implemented in TASO. For EQTL, the data of the expression will be normalized, and those genes with detectable expression will be included in mapping with a matrix EQTL in R. And I will identify EQTLs that are acting in cis or in trans. For TWAS, the TWAS will be performed by associating gene expression and the phenotype directly according to some methods proposed in 2017 using Bayesian statistical methods. And the idea is to combine the data obtained from GWAS and TWAS using the Fisher combined test also proposed previously. Finally, we can construct a network in order to identify the association of different candidate genes from the results from GWAS, EQTL, and TWAS to establish different relationships in the control of quality traits. For the second project, we also have an overview here of the materials and methods from plant material, genotyping, bioinformatic predictions, and effect of and the determination of the effect of mutations. In the plant material will be plants with accumulated self-link generations. In the genotyping and phenotyping, I will use approaches such as a genotype by sequencing and the measure of basic variables in the field. And for bioinformatic predictions, I will use different algorithms. And finally, for the estimation of the effects of mutation, there are different approaches to identify them. So in this case, for the plant material, I will develop a population until F5 generation obtained by single descent method in order to guarantee the selfing process in the plants. For the genotyping, the method is the DNA extraction and the genome complexity reduction and variant calling using genotype by sequencing approach and restriction enzymes previously reported for beans or the test of new ones. In the phenotyping, some variables are the yield-related and physiological traits, such as shoot biomass, seed number, and total seed weight for the analysis. And the effect of deleterious on non-deleterious mutations will be estimated using RR-BLOB as proposed previously using the blobs for phenotypes and the genotypic data. As the research impact, we have the identification of a robust group of genetic elements involved in the regulation of quality traits of common being, understand the role of mutations present in dry bean population, uncovering the genetic processes leading to avenues for the restriction of genetic gain in breeding programs because of deleterious mutations in areas of low recombination in the common bean genome. As expected results, I hope to have a highly inbred population for mutation studies in common bean for different traits and in the future, a set of candidate genes and markers related to the control of quality traits in dry bean, 3 submitted papers from the different projects, and my master degree. Finally, as a schedule, I have some proposed activities here and the date. For example, the research planning and proposal, the first 5 months, mainly from home, now that I am remotely. The establishment of experiments in the field, which is normally made in June. The development of the population for the mutation accumulation study, the pretest for phenotyping quality traits in order to standardize the different techniques and methods, the actual phenotypic data collection, and finally the data analysis, thesis writing, and papers writing will be performed in the last 8 months of the degree. Thanks.
Thank you, Henry. So before we move into the second one, we'll spend a few minutes for questions. You can either write your questions in the chat, and I will ask the question to Henry, or you can use the little reactions button, and I can give you the word.
Can we ask the question directly to the student?
Yeah.
Yes, go ahead.
Okay. It's good, but how many of the cultivars you're going to do this experiment? One type of how many varieties you're going to use?
Oh yeah, I forgot to mention that. The idea for the association studies is using the Mesoamerican panel, which is composed of 280 individuals with different with the variability for these traits.
So in all your experiments, you're using one dry bean variety?
No, this is from the Mesoamerican gene pool. So we have different accessions there with variability in the different traits. It's a panel, it's a composed panel that is used for different traits. It's like the arrangement where you can guarantee that we have the variability for traits, and you can identify how these traits and genotypic data are correlated.
So have you done field
are not
studies?
like domesticated
in your schedule, you had field studies in 2021. How many of these germ clouds you've planted? Have you planted already in the field?
And not yet. I have to plant the plants.
I have a question as well. Just so you know, Valerio, in the reaction button, you can only clap or give a thumbs up. You can't raise your hand. So I guess if someone claps or gives a thumbs up, it means they have a question.
Yeah, it's okay. Oh, sorry, I meant okay, because there is and the participants. Well, yeah, if you give me a thumbs up, I can also unmute you. But I think if you have a question, you can just unmute yourself and ask the question.
Okay.
Because the chat is disabled, Valerio. The chat is disabled.
Is it really? Okay.
Okay, so Henry, hi. Thanks for the presentation. I have just 2 questions. The first one, well, they're actually related. So you're going to be looking at quality traits. So are you planning to look at different environments when it comes to the quality traits, when you look at the phenotyping, or are you going to do just one environment, because the quality traits can be quite affected by the environment?
Yeah, so far I haven't defined that part because I think it would be valuable, as you mentioned, to test different environments and to analyze the relationship and the interaction between the phenotype and the environment. But I think I will, but I haven't defined it yet.
Okay, and my second question is regarding the anti-nutrients.
Yeah.
Or is the concentration of anti-nutrients in beans high, and how does it affect the quality traits of the beans themselves?
Yeah, in the anti-nutrients, we have some factors, for example, that affect the absorption of other nutrients or increase the, for example, phytic acid. Usually when you consume a higher content of this component, the absorption of the nutrients that are beneficial for you is reduced. So the idea is to identify genetic materials with a higher content of nutrients, but a lower content of these anti-nutrient factors. So the idea is to accomplish quality taking into account both, because usually you can find some varieties with a high level of nutrients, but also with phytic acid, for example. So the nutrients you are consuming will want to be absorbed in your body, and we will have malnutrition, for example, in the larger scale.
Okay, thanks.
Okay, you're welcome.
Okay, any other questions?
Hi, it's Marian. I don't have a question, but I just wanted to inquire. I am unable to reach the evaluation form on the My Courses site. It has the folder for the surveys, but there's nothing in there. I'm wondering if I'm the only one or if others.
No, there seem to be others. Okay, I will look into that. So
Okay.
if we can't get it working today, I'll follow up, and then you can fill in your survey. So if there's no other questions, oh, there is a question. Okay, Suha is asking that you need to specify your application, just as a comment,
Okay.
for your analytical experiments.
Okay, thank you.
You have to unmute yourself, Suha. Okay, well, let's move on to our second speaker, is Diljot Kaur.
Thank you.
Thank you, Henry. Diljot obtained her bachelor's in biotechnology from Punjab Agricultural University in India in 2017. And she did then a master's with distinction at the University of Leeds in England, where she was awarded Great Britain Scholarship in conjunction with the British Council. And then she was previously worked on projects in the identification of translocations in wheat using market-assisted selection and recombinant expression of plant catalase to analyze its interaction with another peroxisomal protein. Her aims are to investigate the intricacies of plant insect systems at the molecular level with the goal of contributing towards agricultural improvement. So Diljot, I think you're our co-host, and you should be able to share your presentation now.
I hope you're able to see my presentation.
Yes.
Yes.
I can see it.
Okay, so in this project, we'll be investigating the role of post-translational modifications of various plant proteins and plant-insect interactions. So plants respond to mechanical damage or attack by chewing herbivores by activating jasmonic acid biosynthesis pathway. And this begins with the cleavage of galactolipids in the chloroplast membrane by the action of phospholipase A1 enzyme, which leads to the accumulation of polyunsaturated fatty acids such as linolenic acid. The oxygenation of these fatty acids by the action of an important enzyme, lipoxygenase 2, along with two other enzymes, leads to the formation of OPDA. The OPDA is transported from chloroplast to the peroxisomes, where it undergoes a reduction and 3 cycles of beta oxidation to form jasmonic acid. After mobilization of the jasmonic acid to the cytosol, the enzyme JAR1 catalyzes the conjugation of isoleucine to form a bioactive form of jasmonyl isoleucine. In the normal undamaged plants, when jasmonyl isoleucine levels are low, the JAS repressor proteins remain bound to the MYC2 transcription factors along with its co-repressors NINJA and TPL. This represses the expression of jasmonate responsive genes. However, under stressed conditions, plants elicit a burst of jasmonyl isoleucine accumulation. This triggers the binding of JAS repressor proteins with the SCF-CoA1 E3 ubiquitin multiprotein complexes. So this targets the protein for its degradation by the 26S proteasome. This derepresses the MYC2 transcription factors, which leads to the activated expression of jasmonate responsive genes, which can further lead to production of defensive proteins and metabolites. Spodoptera exigua is a generalist caterpillar species with a life cycle of around 24 days, and the caterpillar labial saliva contains effectors which are known to interfere with jasmonate signaling, and the labial saliva is secreted from a specialized mouth organ, which is called spinneret, as shown in this figure. Lipoxygenase 2 is a very important enzyme which is involved in the initial steps of jasmonic acid biosynthesis. Arabidopsis thaliana has 6 isoforms of lipoxygenase, and lipoxygenase 2 is considered to be the most important isoform in terms of jasmonate signaling. The gene encoding this protein is expressed in the leaves, and the protein is targeted to the chloroplasts after being synthesized in the cytosol. And lipoxygenase 2 reaction is catalyzed around a 5-carbon unit, which has a methylene group in between a pair of double bonds. The oxygen is added onto carbon 13 of this pentadiene moiety, and it results into the formation of a conjugated diene, which is detected at 234 nanometers. So previous research in Bede's laboratory found that this protein lipoxygenase 2 remains constitutively phosphorylated at position 600 in case of the undamaged plants or the plants which are attacked by caterpillars with normal salivary secretions. However, under wounded conditions such as mechanical damage or attacked by caterpillars with impaired salivary secretions, a putative phosphatase is believed to dephosphorylate this enzyme. Now, post-translational modifications can have various effects on the protein. It can regulate its activity by regulating kinetic parameters. It can affect its interaction with other proteins, its localization, and can also target it for its degradation by the proteasome. Based on Bede's findings, which suggest that the lipoxygenase 2 remains constitutively phosphorylated, we hypothesize that the enzyme is present at a low basal activity in this condition, which leads to less induced defense responses. However, in case of wounded conditions, we suspect the enzyme to be activated and leading to increased jasmonate responsive defenses. So our first objective is to evaluate the role of LOX2 phosphorylation on enzymes activity in vitro. And this will be done by characterization of various kinetic parameters of the enzyme by spectrophotometric analysis. So for this, we have started this experiment by recombinantly expressing the protein in vitro in a bacterial expression vector. And the purified protein was analyzed on STS polyacrylamide gel electrophoresis at an expected size of 98.7 kilodaltons. To analyze the kinetic parameters, we carried out spectrophotometric assays for which we loaded the assay mix containing our enzyme onto the linoleic acid substrate, and the increase in absorbance was measured at 234 nanometers for 15 minutes in a spectrophotometer. Now, most of the enzymes follow Michaelis-Menten kinetics, and according to this, With increase in substrate concentration, the rate of reaction reaches its maximum velocity. And in our results, we found that the enzyme follows a sigmoidal pattern rather than a hyperbola, which means that the enzyme does not follow Michaelis-Menten kinetics and instead has a cooperative behavior. And this has never been found earlier for lipoxygenase enzymes. To analyze the kinetic parameters and pH optima, we carried out the assays at a range of pH from pH 7 to 8.8. And we found that at the neutral pH, the enzyme follows more of a sigmoidal pattern, whereas at the alkaline pH, it tends to shift towards the hyperbolic form, but it still follows the sigmoidal pattern. So the first kinetic parameter that we analyzed was maximum velocity, and we found that the Vmax is highest at a pH of 8.2. And the second parameter was Hill constant, which corresponds to the degree of cooperativity. And we found that the enzyme is highly cooperative at neutral pH. The third kinetic parameter was S0.5, which corresponds to the affinity of the enzyme for its substrate. So lower the value of S0.5 means high affinity of the enzyme, and we found that the enzyme has highest affinity at a neutral pH. These different kinetic parameters will be compared to the different LOX2 mutants, which are available at Jean Rival's lab. And these include a non-phosphorylatable mutant, where the serine at position 600 has been mutated to alanine, and two other phosphomimic mutants, where the serine has been mutated to aspartate and methionine. This is the image taken from a former master student in our lab, and it shows a Western blot analysis of a phosphomimic mutant showing degradation. So we hypothesized that LOX2 phosphorylation might act as a target for its degradation, which led to our second objective, which is to analyze the effect of phosphorylation of LOX2 on enzyme stability. For this, we'll be generating Arabidopsis cell suspension cultures for different genotypes of lipoxygenase 2. This will be followed by its treatment in the absence and presence of a proteasome inhibitor, which is MG132. This will be followed by extraction of the protein, its separation on STS polyacrylamide gel electrophoresis, and its transfer on a nitrocellulose membrane to analyze the patterns of degradation by detecting with the LOX2 anticlonal polyclonal antibody. The whole of this experiment will also be repeated in the presence of methyl jasmonate to induce defense responses. So this will help us to investigate the stability of the enzyme in response to wounding. This is how we expect our proteins to migrate on the gel. So if phosphorylation acts as a signal for the degradation of the protein, we expect to see a degraded band in each case except for the non-phosphorylatable alanine mutant. And in case of the presence of methyl jasmonate, we expect the proteins to be dephosphorylated, and hence the result will be a sharp and clear band, except for the phosphomimic mutants, which will still be behaving as phosphorylated serines and showing degradation. So our next hypothesis is that the effectors in the labial saliva allows the protein to remain in its phosphorylated state and hence circumvent the jasmonate-mediated defense responses. However, in the absence of the labial saliva, we suspect that the enzyme will be dephosphorylated by a putative phosphatase, and it will result into an increased jasmonate-mediated defense response. The third objective is to analyze the role of phosphorylation in plant-insect interactions in vivo. For this, we'll be using Arabidopsis transgenic lines, and these have been prepared by a previous master student in our lab, and she prepared these by transforming various LOX2 mutants into a mutant background line. which was provided by Dr. Edward Farmer. And we'll be using two different populations of the caterpillars. And the first population is the normal caterpillars with intact salivary secretions. And the second one are cauterized caterpillars, where the spinneret has been cauterized by using a hot probe. So the wild type Arabidopsis line are mutant background, and the other transgenic lines will be subjected to 4 different treatments, which includes unwounded, mechanically wounded, plants attacked by normal caterpillars, and the plants attacked by cauterized caterpillars. And these differences in gene expression will be analyzed by quantitative reverse transcription PCR. And for the phytohormone and glucosinolate levels, the samples will be sent to our collaborators in Germany, and the experiments will be repeated for 4 times at least, and the significant differences will be analyzed by 2-factor analysis variance. The another protein which I'll be working on is an abscisic acid responsive element binding factor, and it belongs to the family of basic leucine zipper transcription factors, and it executes transcriptional regulation of various abscisic acid related genes. Beats laboratory also found that this protein is post-translationally phosphorylated at serine 431 and nitrosylated at cysteine 420. So we hypothesize that these post-translational modifications results into its activation and enhanced binding to the ABRE motifs. So our objective 4th will be to characterize the effects of these post-translational modifications on its recognition and interaction with the consensus sequence ABRE. So for this, we'll be expressing the different ABF3 variants in vitro, and these includes mutations where the serine 431 has been mutated to aspartate and alanine to mimic phosphorylation and as a non-phosphorylatable mutant, and cysteine 420 have been mutated to tryptophan and serine as a nitrosylation mimic and non-nitrosylatable mutant respectively. This will be followed by carrying out electrophoretic mobility shift assay, which involves labeling of the DNA probe, its incubation with the purified protein fractions and gel electrophoresis and visualization. So this figure shows a typical setup of an EMSA reaction, where in lane 1, we'll have only our labeled probe, which will be found as demonstrated as a single band on the gel. The second lane will contain our protein, and if they bind each other, the complex will migrate on the gel with a higher molecular weight, and hence it will be seen as a band shift. The third lane will be a competition reaction, where an unlabeled DNA sequence will compete in binding with the protein and will result in a disappearance of the band. The fourth lane is a non-competition reaction, so it will not compete in binding to the protein, and it will be seen as a band shift, just as in lane 2. And lane 5 will have an antibody, which will result into even higher molecular weight of the complex and will be seen as a super shift. So until now, we have been successfully able to express and purify the active form of lipoxygenase 2 protein, and we have determined various kinetic parameters. And this project will help us to understand the role of post-translational modifications in regulating various plant defenses, and to understand the role of effectors in interfering with plant processes, which will further lead to help us develop crop protection programs. In the end, I would like to thank my supervisors, Jackie Weed and Jean Revoil, my committee members, and Sonia for technical assistance, my lab members for their constant support and encouragement. Ready to take any questions? Thanks.
Okay, questions for Diljot. Thank you so much.
I have one question. Hi, Diljot. Thanks for the presentation. I'm not an expert in herbivory, but in one of your objective, I think it was objective 3, you mentioned that you're going to compare having caterpillars that are fully functional, and then you're going to have some that have been categorized.
Yes.
So do you think that those who have been categorized, it's going to affect the way they're feeding on the plant? And is that why it would have an effect on the plant reaction?
So they are not going to affect the feeding of the caterpillars, but the one which are cauterized won't be able to secrete like their labile saliva. So the labile saliva actually has effectors which can suppress the defenses of the plants. So we are going to compare them with the normal caterpillars, which will be able to suppress. So that's a way of testing gene expression and comparing them between the two populations.
Okay, so even though that you're categorizing them, they're going to still be able to feed normally as any other caterpillar.
Yes.
Okay, thanks.
Some questions here. Does the saliva of all caterpillar species induce these effects?
They don't induce it, but they have like effectors which can suppress them. For chewing herbivores, not many of the effectors like have been known, so their targets are not known. But for like phloem or sap-sucking insects, there are more effectors that have been known and their mechanism of how they suppress the defenses.
I see. Just an announcement. So yes, I think the survey for anybody that is regularly registered in the course, it should be working. Let me know if it's not. There are a few people that are registered as guests because they have taken it in the past, and so so I think for those people it won't be working. But do let me know if you are registered for grade and you are not able to complete the survey. They should be working now. Any other questions for Diljot? Well, if we have no more questions, then thank you to Henry and Diljot for your presentations, and wish you the best.
Thanks.
Thank you.
Valerio?
Yeah.
So did you see my last comment?